Each of us is a symphony, our genes the musicians. And while each is important, without the control of a conductor, the piece ultimately falls apart. Our epigenome is that conductor. I fucking lost it. It's totally gone. Do you mind if I start again?
Take it back and start again.
Okay. Each of us is a symphony, our genes the musicians. And while each is important in its own right, without the control of a conductor, the piece ultimately falls apart. Our epigenome is that conductor, a key mechanism in controlling gene expression. See, gene expression is not an issue of just on or off, and it's not the same across all cells. And this gives rise to the variety of behaviours we find in normal and disease cells. Indeed, epigenetic dysregulation has now been linked to, amongst other things, cancers. And so synthetic control of the epigenome presents a valuable tool not only in unravelling the mysteries of the cell, but in potential therapeutic applications. Now, this is not a new concept, but up until fairly recently, the methods used have presented significant caveats. Enter CRISPR, a system found naturally in bacteria as immunity. It comprises two main components, a short guide RNA and the Cas9 protein. Now, the guide RNA recognises DNA using an extremely simple system. And so this presents a straightforward and highly tractable method of DNA targeting. By fusing an effector, that's an activator or repressor, to a deactivated d-Cas9, we can target an effect to just about anywhere we want. But Cas9 has been shown to bind off-target at multiple sites across the human genome. It's the epigenetic equivalent of our conductor randomly waving his arms around. And so two main methods have come forward to combat this. Further mutations to the d-Cas9 to create an enhanced specificity version, and modifications to the overall system, which involve bringing in the effectors via the guide rather than the Cas9. This is known as SAM. And the SAM system has been used to remarkable effect to upregulate many targets. But repression has been largely ignored, despite representing a powerful tool. And so we address two questions. Do current methods truly overcome the issues of specificity? And can we use a repressive system to fully kill a target? Now, while we've been able to recapitulate results in upregulating a target, unsurprisingly, we found repression to be less potent. And this is because the pathways involved in repression take longer to process than activation. And in our current method, each part of the system is only available in the cell for a short amount of time. So to combat this, we have to insert each element, that's the Cas9, the effector and the guide, into the genome of the target cell, giving us stable long-term expression. And with stable expression of a repressive system, we open the door to a whole host of possibilities. Mapping regulatory sequences to their targets, looking at the sequences that are involved in the 3D structure of DNA, not to mention examining gene function without the requirement of the lengthy process of gene knockout. And so, in the process of refining the symphony of the cell, what my work represents is one step forward in the journey from Bieber to Beethoven.